Assalamualaikum and good day everyone. We are from Group 3 Biotechnology. We'll present our title, which is Triticale and Sorghum as feedstock for bioethanol production via consolidated bioprocessing. Introduction. Implementing a circular bioeconomy is vital for combating climate change, with biofuels like bioethanol offering a sustainability alternative to fossil fuels. In South Africa, there's a growing focus on utilizing triticale and waxy grain sorghum as raw starch feedstock for bioethanol production, aiming to enhance sustainability while addressing food security concerns. Our study aims to evaluate triticale and waxy grain sorghum flour as raw starch feedstocks for bioethanol production and assess different fermentation conditions to optimize starch to ethanol conversion. However, challenges persist in developing economically viable methods for bioethanol production using these feedstocks, particularly in optimizing fermentation condition and understanding the physical changes in starch granules during enzyme treatment. Thus, detailed investigation and optimization are crucial for effective utilization of these grains in consolidated bioprocessing.
Today, we are exploring the fascinating potential of triticale and sorghum for bioethanol production. The literature highlights sorghum's high sugar content and adaptability with studies like Batok showcasing the remarkable Sorgo 506 variety. Meanwhile, triticale, a resilient wheat-rye hybrid, demonstrates impressive bioethanol yields under optimal farming practices as seen in Kilcoca. Together, these studies illuminate the path towards sustainable biofuel solutions, leveraging nature's bounty for our energy needs.
Starting with the strains, media, and cultivation, as stated in the flowchart, to industrial Saccharomyces cerevisiae we use, namely parental S. cerevisiae ethanol version 1, ER, and recombinant ERT12 strain. The yeast strain will remain fixed on YDP agar and cultured in YDP broth at 30 degrees Celsius. For the substrate preparation and analysis, triticale grain and sweet waxy sorghum grains were obtained. The grains were then grounded with a hammer mill with a 1.25 mm sieve. Fractions smaller than 0.05 mm would be classified as flour, and the other were classified as bran fraction. The starch content of both flour and bran were determined according to the acetate buffer method by using a commercial alpha amylase. Glucose concentrations were quantified using D-glucose assay kit, and the protein, cellulose, hemicellulose, lignin, and ash contents were quantified using international standard methods. Moving on to the starch fermentations, pre-cultures of both S. cerevisiae were prepared in a 50 ml YDP medium in 250 ml Erlenmeyer flask at 30 degrees Celsius with shaking at 200 rpm. Small-scale fermentations were conducted in 120 ml serum bottles sealed with rubber stopper for anaerobic conditions. It contains 10% volume by volume inocular, 20% weight by volume substrate, and either YDP or RO water added to reach 100 ml. Antibiotic ampicillin and streptomycin were also added. Then the cultures were incubated for 168 hours at 360 rpm and 30 degrees Celsius for HPLC. Continuing from starch fermentations, HPLC analysis was conducted. Liquid fractions from sample were diluted and filtered with nylon string filters. The ethanol conversion efficiency can be calculated using the formula provided in the slide. The analysis must undergo 3 repeats. Statistical analysis was performed using t-test, and p-values less than 0.05 were considered significantly different.
Triticale and sorghum in both fraction, bran and flour, contain starch, protein, cellulose, hemicellulose, lignin, ash, and lipid, which peptide and amino acid will serve as nitrogen source for Saccharomyces cerevisiae growth. High starch content will yield higher ethanol production. The starch content shows relatively higher in flour fraction for both sorghum 79% and triticale 73.5%. Triticale and sorghum flour have similar protein content to cornstarch. Protein content in triticale shows higher protein content than sorghum. High protein content imply a lower starch content and also ethanol yield, but more free amino nitrogen available to the yeast.
Next, we move on to the comparative starch fermentations. Fermentations using 20% triticale and sorghum were carried out with SSF and CBD methods, both with and without added nitrogen. These were compared based on ethanol concentrations and productivity as seen in Figure 1. Using water in the fermentation both led to higher ethanol concentrations and better conversion efficiencies from both flour types compared to using YPD broth within the first 96 hours. However, CBD worked better on sorghum while SSF was more effective on triticale flour. After 168 hours, YPD and water produced similar results for CBD and SSF on triticale flour, but CBD remained more effective than SSF on sorghum flour. The results showed that adding nitrogen was unnecessary for raw starch CBD using triticale and sorghum. Water that is commonly used to prepare mash in dry grind ethanol plants works well for starch CBD fermentation without harming ethanol production or yeast growth.
The recombinant ERT12 strain could produce high ethanol conversion efficiency in SCBP. Sublimited CBP only investigated for the water configurations as it yielded better result than YDP media. Sublimated CBP increased ethanol conversion efficiency to 86 and 97% at 96 hours respectively, higher compared to SSF and CBP, and decreased the fermentation time closer to the target of lesser than 72 hours for industrial processes. Similar case with sorghum. Supplemented CBP improved ethanol production over the first 72 hours. For fraction, both reaching almost 99% ethanol conversion efficiency after 96 hours. It is confirmed that the GSHE supplementations could eliminate the lag phase, enhance ethanol productivity for high yields in less time, at the same time reduce operating costs.
And now we move on to the triticale versus sorghum as CBD feedstock. The CBD approach yielded significantly more ethanol from sorghum bran and flour than the corresponding SSF fermentations. This can be seen in figure 1b and 2b. The significant ethanol production indicates that the amylase from the ERT12 strain worked better on sorghum than the Stojan 002 cocktail used in SSF. However, Stojan 002 was more effective at breaking down triticale starch. Sorghum grain has abundant asparagine and glutamine, which yeasts are readily absorbed during early fermentation. In contrast, triticale has lower levels of amino acids that yeast can easily assimilate.
Conclusion, the amylolytic ERT12 strain achieved ethanol conversion efficiency at 196% from sorghum after 96 hours and triticale flour after 168 hours respectively. Supplementation with 10% of recommended enzyme loading significantly reduced the fermentation time. Ethanol conversion efficiencies reached 92% for sorghum and 72% for triticale flour after 48 hours. It was proven that SSF approach was better for triticale substrate and sorghum substrate were ideal candidates for a CBP-based approach. Both substrates were fitting starch feedstocks for fermentation with amylolytic strain.